Okay, so I think we got almost everybody, maybe one person absent. All right, so again, we're going to start with the Canvas page, starting with the Canvas page that says DNA and protein structure and function of DNA. That's where we're starting today. I'm going to share with you in just a second, as soon as I do attendance. So again, please log into Canvas. Please log into Canvas, and because that's where we're going to start. Okay. uh, okay. Got one on the way and one absent. There she is. All right. Some of this vocabulary we've done before. When we did our chart at the beginning of the semester, we did a macromolecule chart. And if you find that, you might want to pull that out. It had the monomer for nucleic acid. So that's what we're looking at today. We need to talk about the polymer. We need to talk about nucleotides and what it's made of. So refresh your memory with that as well. And like I said, give me 1 second to finish this attendance, and then I will share. Okay, everybody should be on Canvas, and everybody's not on Canvas. Really? Okay. All right, so we're going to start at the top, and I'm going to go ahead and go through, just to go through the DNA timeline. So this is just a historic background of the discovery of DNA. And then when we do the Nearpod, it's going to kind of pick up where this one leaves off. So this one is going to leave off where the Nobel Prize winners for the discovery of the structure of DNA. But a lot of research went into um, discovering what DNA actually is. Actually, if I can get So all we have to do with this, oops, not that one, this one. I don't know why my screen is blank. There we go. All right, so if you haven't already done it, we're going to do it together.
Miescher discovers nuclein. Friedrich Miescher was the first scientist to isolate nucleic acids from cells. He did this by collecting the white blood cells from the dressings of wounded soldiers. He found that this nuclein molecule, located in the nucleus, contained phosphorus, carbon, hydrogen, oxygen, and nitrogen. Nuclein was later named nucleic acid. Miescher and other colleagues at the time did not realize that nuclein was the molecule of heredity.
All right, so that's at the beginning. So when we first started talking about molecules, we looked at macromolecules were made up of carbon, hydrogen, oxygen, and nitrogen. So they have discovered, and we included nucleic acids as part of that bunch. So this is where they discovered that way back in 1969. Let me say
Griffith
1869.
mice and DNA transformation. Frederick Griffith conducted experiments with two related strains of bacteria. Rough coat bacteria did not cause pneumonia in mice. You can see this in the first column of the diagram. Smooth coat bacteria did cause pneumonia in mice, and the mice died. The reason for this is that the smooth coat actually protects the bacteria from the mouse's immune system. Interestingly, Griffith took this experiment 2 steps further. He then decided that he would try to kill the smooth strain of bacteria first before injecting it into mice. He did this in the third column of the picture. The mouse lived. Finally, Griffith decided to mix both the killed smooth strain and the rough strain together and culture them for a few days before injecting them into the mice. When he did this, he found that the mice died. The conclusion that Griffith made was that transformation had occurred. Some unknown chemical in the dead cells was transforming the harmless rough cells into dangerous smooth cells. We now know that this chemical was the DNA from the heat-killed cells that was not destroyed. Bacteria can actually take up the DNA from their environment and use it to make smooth coats, making them more deadly.
All right, and we've been noticing this too with the medicines that we have today. They adapt. So they change with the body temperature. So transformation is something that DNA does. So we'll talk about that later.
Avery, MacLeod, and McCarty. These scientists purified and tested all the chemical components in the smooth and the rough bacteria from Griffith's experiment to determine which component caused the transformation. Their conclusion was that this was likely DNA, but at the time very little was known about the composition or the functions of DNA. In fact, most scientists still thought that proteins were the hereditary or the instructional material for cells. Chargaff's rules. Chargaff studied the molecule of DNA and determined that there was always the same amount of the chemical adenine as there was thymine, as well as the same amount of cytosine as there was guanine within the DNA molecule. His conclusion was that adenine and thymine must form pairs, and then the same would be true of cytosine and guanine.
All right, so we're going to come back to that. When we do the Nearpod, you're going to have to remember this rule. And so just remember this one.
Hershey, Chase, and blenders. Hershey and Chase experimented to find out if the transforming principle from Griffith's experiments was DNA or if it was protein. Hershey and Chase used a virus called a bacteriophage that infects bacteria. They knew that bacteriophages were made of 2 things. One was DNA, which contained phosphate, and the second was protein, which has sulfur. Which molecule would enter the cell to make more viruses? They labeled the phosphorus and sulfur, each with radioactivity, and observed what happened in 2 experiments. Look at the diagram. Since the protein coat of viruses contains sulfur, the viruses were treated with radioactive sulfur that would bind to the protein but not DNA, and be easily detected with special equipment. This is shown on the left-hand side of the diagram. On the right-hand side, you can see that since DNA has phosphate molecules and not sulfur, the viruses were also treated with radioactive phosphate that would bind to the DNA but not the protein. The treated viruses both infected the cells, and then the cell parts were broken apart in, of all things, a blender. They separated the pieces into groups and determined that the DNA with phosphate was the material that was being injected into cells, and not the sulfur-containing protein. Radioactive sulfur was found on the surface of bacterial cells, and radioactive phosphate was found inside the bacterial cells. The conclusion here was that only the genetic material, DNA, entered the cells and caused transformations.
Okay, we're going to talk about that too when we talk about the role, the structure and role of DNA and RNA. Who can go in and what can go in and what can go out are going to be important. My last one.
In 1953, the structure of the DNA molecule was revealed. James Watson, Francis Crick, and Maurice Wilkins examined an amazing photograph made by Rosalind Franklin, a colleague of Wilkins, using a technique that she developed called X-ray crystallography. Together with this information and the studies of many other scientists, Watson and Crick deduced a model for the structure of DNA. For this, they all received the Nobel Prize. Their model for the DNA molecule consisted of two nucleotide chains that wrap around each other to form a double spiral. This shape was called a double helix.
Okay. So, like I said, the near part we're going to do is going to pick up from where they left off. We are not going to do this right here, but you should read it. But that's all of this is going to be in the near part as well. So we are going to, when we finish the near part, we're going to come back and start about right here. So again, I need everybody's participation. So once I give you the code, I need you to log in. All of your login information is recorded. So participation, all that good stuff is, I need a new one. So please make sure that you're logged in and you're answering questions. All right, so the login is in the chat. Give you a few minutes for everybody to log in. All right, so again, as always with the Nearpod, it stops along the way and it asks you questions, and I will give you a minute to answer the question. We'll review your answers, but I do expect everybody to have an answer.
This is DNA. This is the molecule we use to store our genetic information. DNA contains the instructions needed for an organism to develop, survive, and reproduce. If you want to understand how DNA works, the first step is to learn about its structure. The structure of a molecule determines its function, so making sense of the structure is essential for you to understand how DNA can do its job.
All right, so I'll give you a minute to answer the question. What do you already know? So we just went through a little bit history. Like I said, at the beginning of the course, we looked at it as nucleotides as a macromolecule. So you know a little something. So everybody should take a minute to write a sentence about what do you already know about DNA. Let's keep rolling on. Landon, are you with us? You look like you're thinking too hard. All right, so like I said, this unit has a lot of vocabulary, has a lot of technique. We're going to do a lab on Wednesday. So try to make sure that you are taking notes, but keeping up. All right, so let's see what you got to say. All right. Zyla, no answer. I need an answer from everybody. Where are you? What you doing? All right, the structure, Maxwell, I'm not sure I understand what that means. Okay, that's good. Thank you. The polymer. Okay. Thank you, Ariana. It makes up who we are. That's good. Has a double helix. That's good. Okay. Okay. All right. Good, 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 good. I know it makes up. Okay. Thank you. Thank you. Good answers. Good answers.
In April of 1953, James Watson and Francis Crick changed the world of biology forever with a landmark paper published in the journal Nature. It was just 1 page long. They reported they had determined the structure of DNA was an anti-parallel double helix. How did Watson and Crick figure this out? They didn't do any experiments. Instead, they built models out of tin and wire that matched measurements derived from other scientists, including biochemical data from Erwin Chargaff and X-ray diffraction data obtained by Maurice Wilkins, Rosalind Franklin, and Raymond Gosling under the direction of John Randall. You may have tried making your own DNA molecule using a chemistry molecular modeling kit. Today, we're building models of DNA using a 3D printing pen. MorphPen was kind enough to send a 3D printing pen to Socratica for us to test out. So of course, we're making DNA with it, obviously. You can make whatever you want with your 3D pen. Single-stranded DNA is a polymer of nucleotides, made up of repeating units of sugar, phosphate, and nitrogenous base.
All right. So, so which of the following units are joined repeatedly in a nucleotide? Now, there actually are 2 answers here. So you pick 2, because so this it'll show up as like kind of like 50-50. Or 3 answers. Actually, this is the one with the 3 answers. Okay. All right, somebody's not answering. When it tells me no answer, that means somebody didn't answer. But yes, the repeating units, when we talked about this earlier too, when we talked about a polymer is many units, remember? Poly is many units. The mer is the basic structure. We had monomers and polymers. And in the DNA molecule, the nucleotide unit is made up of a nitrogen base, a phosphate unit, and a sugar unit, repeated and connected. Okay. All right, we still had somebody who didn't answer. Remember, it tells me who doesn't answer and who does.
In double-stranded DNA, 2 polynucleotides are attached at their complementary bases, zipping the 2 strands together with hydrogen bonds. Keep in mind, most DNA molecules are very, very long, thousands or even millions of base pairs long. We're just going to make a very short stretch of double-stranded DNA as an illustration. DNA looks like a twisted ladder. The sides of the ladder are made of sugars and phosphate groups. These repeat over and over. Sugar, phosphate, sugar, phosphate. This is the backbone of DNA.
Okay, this is the one I said has two answers. All right, so if you select both answers, the pie should look like 50-50. And again, somebody is not participating. Because I shouldn't see 11% no answer. That little gray spot should disappear if everybody's answering. Thank you. That's why you're here, to participate, not to sit back and stare at the screen. All right. So, okay, good. Oh, finally, she comes to class. Okay. All righty. So, repeatedly, so when we talk about DNA being like a ladder, the sides of the ladder are what we call the backbone, okay? And so the sides of the ladder are made up of two units, the sugar and the phosphate. Next, we're going to talk about the rungs of the ladder, the steps of the ladder.
Let's first make a backbone using our 3D pen. We're actually going to need two of these, one for each side of the ladder. Now, let's look at the rungs of the ladder, which is really the business end of DNA. These rungs of the DNA ladder are made up of 4 different kinds of nitrogenous bases: guanine, adenine, thymine, and cytosine, abbreviated GATC. The order of these bases is how information is stored in DNA. Think of it like the order of letters in a word, and the order of words in a sentence. Guanine always pairs with cytosine. Adenine always pairs with thymine. These are known as Chargaff's rules, in honor of Erwin Chargaff, who determined this biochemically.
Okay, so I told you from the notes on from Canvas that would come back up. So go ahead and answer that question. And this is, like she said, this is the business end. This is one of the most important things that you have to know about DNA. Who matches with who? Which base matches with which base? Oh, okay. Hold on. Mm-hmm. All right, and again, I expect everybody to answer. And most of the time when these questions come up, right before the question comes up, they give you the answer. So again, they're just checking to see, are you paying attention? But this was one of the, Chargaff was one of the scientists that were in our timeline, and he has a rule. Oops, okay, hold on. Let me go back. Didn't mean to do that. That's what I meant to do. All right, Maxwell. I hope you're working on an answer. All right, so the amount of guanine should equal the amount of thymine. That's half of it. That's good. Rule states that in a double, you keep on going. Keep on going. DNA molecule equals, okay, there we go. Natalie has it. So I think, then, okay. And we don't want, Piper, we don't want to say equal. They pair. So this is about pairing. So yes, there's going to be an equal amount, but the important part is that they pair together. So adenine is always going to pair with thymine. Guanine is always going to pair with cytosine. And because they're always going to be paired, there's always going to be an equal amount of those in, they're going to be equal to each other. And okay, still no answers up here, folks. Let's go, let's go. But I am going to move on. I am going to move on.
Notice that there are 3 hydrogen bonds holding G and C together, while there are 2 hydrogen bonds holding A and T together. Let's take a closer look at these components. The 5-carbon sugar in DNA is deoxyribose. Deoxy meaning it's missing an oxygen compared to regular ribose. The phosphate group looks like this. This is where it's attached to deoxyribose. The phosphate groups are acidic. Think of them like little acidic flags hanging off the structure. This is what gives DNA its acidic nature when in solution in water. Now how about those nitrogenous bases? They're called that because they're literally basic as opposed to acidic, and they all contain nitrogen. 2 of them, adenine and guanine, look like this, with 2 fused rings. These bases are called purines. The other 2, cytosine and thymine, look like this, with only 1 ring. These bases are called pyrimidines.
Okay. And again, this one will have 2 answers. Because 2 of them are pyrimidines, 2 of them are purines. Okay, and I think you need to write this one down. So it's up, so the pyrimidines, this one just says purines. So the purines are A and G, but they're not the ones that match. Okay. A and G are purines, and C and T, or T and C, are your pyrimidines. Okay, so this pie should be half blue, half pink, because both of those answers are correct.
Since adenine always pairs with thymine and guanine always pairs with cytosine, you can also say that a purine always pairs with a pyrimidine. 1 unit or monomer of the DNA polymer is called a nucleotide. This is 1 sugar, 1 phosphate, and 1 nitrogenous base. In the case of DNA, they're also called deoxyribonucleotides, as opposed to in RNA where they're called ribonucleotides because they contain ribose instead of deoxyribose. Nucleotides are joined together by covalent bonds. The phosphate group of one nucleotide is linked to the hydroxyl group on the sugar of the next nucleotide. These are known as phosphodiester linkages. Here's a technical note. Biochemists have a particular way of labeling the atoms in DNA so they know what part of the structure they're referring to. This is an important shorthand. So you can just say something's happening at C5, for example, rather than having to point it out on a diagram. They start with labeling the skeleton, the atoms in the ring area, of the nitrogenous base. You can see here these atoms labeled 1 through 6, or over here labeled 1 through 9. Then, to label the skeleton of the sugar, they don't want to use the same numbers that they used for the base because that would be confusing, so they label them 1', 2', 3', 4', and 5'. The 5' carbon is this one that sticks up from the ring. This is where the notation 5' to 3' comes from. You may have heard that phrase, DNA runs in the 5' to 3' direction. That's talking about one of the two strands. You can see the sides of the ladder run in opposite directions. Remember, it's an anti-parallel double helix. On one end of a DNA strand, there's a phosphate group attached to the 5' carbon, and the other end of that strand has an OH group on the 3' carbon. We say this side runs in the 5' to 3' direction because of the labeling of the atoms. The other side of the ladder runs in the 3' to 5' direction. This is going to be important to remember later when we study how information is stored in DNA. One side is the coding strand or sense strand, the 5' to 3' side. The other side is called the anti-coding strand or antisense strand. Because of complementary pairing, if we know the sequence of one strand, we know the sequence of the other strand. It's kind of like a photograph and the photographic negative.
Okay, anti-parallel. So we want to explain, remember something is again. So what do they mean by anti-parallel? Parallel, that you got two runs of your ladder. And what do they mean by anti-parallel? Remember, everybody should be giving an answer. Okay. I know, folks, you're still working on it, right? Because I shouldn't see any blanks. All right, that's two strands of DNA. Thank you, Landon. Good answer. When two strands run in opposite direction. And basically, that is the answer, folks. You have two strands of DNA and they run in opposite directions. So anti-parallel, they are lined up with each other, and that's going to be important because we have the base pairing, but they run opposite directions. And so if you're looking at the 5, as we Piper put in here, the 5', 5 to 5' to 3', and then the other one is going in the opposite direction, but they still have matching base pairs. It's just that one starts at the top and the other one starts at the bottom. This is going to be important tomorrow when you do your lab. Yes, tomorrow is Wednesday, because you're going to be asked to do a matching. And one of the things you're going to have to mean was when you start matching, you're going to have to start where you make one strand, you're going to have to turn around and make the other one from the bottom. So that will be important there. All right, Malik, are you with us? Come on, come on, come on, come on. All right, moving on. And
Watson and Crick, rather cheekily, said at the end of their paper that the structure of DNA suggested a mechanism of replication. And they were right. But we'll talk about DNA replication another day. That's a very long, complicated, and serious story. Back to the DNA structure. Remember we said Watson and Crick were guided by X-ray diffraction data. That's how they knew the spacing of the components of DNA. Their physical model had to match the values derived from the X-ray photographs from Gosling, Franklin, and Wilkins. This is what they determined. The radius of DNA is 1 nanometer. This is what you get if G pairs with C and A pairs with T. So this also satisfies Chargaff's rules. If you don't pair G with C and A with T, you get a strand that's either too fat or too skinny to match the X-ray diffraction data. The two polynucleotide strands wrap around a central axis to form a right-handed helix. The helix makes one full turn every 3.4 nanometers, and there's a 0.34 nanometer distance between nitrogenous bases. This means there are 10 layers of base pairs, or rungs, per turn of the helix.
And she just gave you the answer. All right, good job. Yes, there's 10 between each set, and that's what makes, before it starts to twist again, you have 10 units.
Notice there are two different kinds of grooves made as a result of the twisting of the helix. In one groove, the backbones are far apart, and that's known as the major groove. Then we see here where the backbones are close together, and that's called the minor groove. This plays a significant functional role because there are specific DNA binding proteins that will only bind at certain locations, and some of them bind at the minor groove and others bind only at the major groove. Now that you know all about the structure of DNA, you'll be prepared to learn about its function. I have to tell you, this is a huge subject. Really, there's an entire field of biology devoted to the function of DNA and other biological molecules, called molecular biology. I personally am a molecular biologist, so I plan on guiding you through that study.
All right, so take a minute to just jot down something, anything that you might be curious about that we will probably answer as we go through, because we've just started talking about DNA. But she said a lot. One of the things she said earlier is, think about it, when we make a word, we have 26 letters to the alphabet, and we can turn those any letters in any arrangement, and they make different words. In this case, we only have 4 bases, but the order that we put those bases in will determine the code that we write for the protein that we want. So 4 bases are going to be what we're going to start talking about codon. Okay. I say just jot down anything. I don't want a blank next to your name, so just jot down something, something, something about DNA. What are you curious? What did she say that made you want to go, I wonder what it's going to do? All right, so I'll give you another minute to do that. All right, then we're going to switch back to Canvas. All right, I'm going to stop sharing for a minute. All right, so I'm going to close out the Nearpod. The rest of it, she was just going to advertise that you should join her. You uh, subscribe and join and ask more questions. All right, so I'm closing out that Nearpod. Hopefully you answered the questions. All right, so now we're back in Canvas. So again, if you go through these activities, they basically are a review, not that one, a review of what we just did in the Nearpod. Okay, so we have a double helix. We know that they are made of make up the chromosomes. We have what the DNA structure is. It's the sugar, and in this case, the sugar is deoxyribose, and deoxy because it's missing an oxygen, as opposed to ribose sugar, which we're going to have in RNA. So when we're going to eventually have a list of comparing RNA and DNA in a few minutes, and one of the differences is deoxyribose sugar. They're both made of sugar, a phosphate, and a base, but the DNA has the deoxyribose, which is missing an oxygen. Okay. All right. So you need to know what a nucleotide is made up of. You need to know that there are 4 bases. You need to know which bases pair with which bases. And you need to know that it's a rule. It's the base pair rule. Okie dokie. This one is basically, if you go through it, it's just going to rotate the DNA molecule and give you different views of the double helix. All right, so I want you to look at this. Now, this is what you're going to have to do tomorrow. So what they've given you here are a chain of bases, and you're going to get this activity a lot as we go through and start talking about protein synthesis. So right now we're just constructing a DNA molecule, but being able to do this and read this is going to be something that will be important when we do protein synthesis. So it says if you have ATGGCA, you need a complementary strand. So take a minute to write those down on a piece of paper. And again, this is practice. Again, write down and you don't have to write the whole thing. Only you're writing is GCA. All right, so complementary means what is going to match. Every time you have an A, what is going to match. Every time you have a T, what is going to match, and so on. All right, so what the answer that they give you is this. And I want you to write this underneath the letters you just wrote because they match. You're not writing it side by side like it's a sentence. Write this row underneath the row that you just wrote. So in other words, your T should be underneath your A should be underneath the T, both of the C's should be underneath both G's, the G should be underneath the C, and the T should be underneath the A. Okay, indicating base pairing, that they are complementary to each other. All right, so again, tomorrow in the activity that you're going to do, you're going to create double-stranded DNA, and you're going to have to be able to complementary match the 2 strands together. All right, so now we're going to look at what is RNA. One of the other important things about DNA is it's found in the nucleus. It never leaves the nucleus. Sometimes we call the nucleus the brains of the cell, or the boss's office. But this boss never comes out of his office. He has all the blueprints. He has all the information for making whatever needs to go on in the cell, but he never comes out the office. So because he doesn't come out of the office, to get the information out of the office, he needs a partner. So that's where RNA comes in. So what we want to look at is what's the difference between RNA and DNA? So you might want to jot these things down because this is always a test question. So just looking at it off the top, you see RNA has a single strand and DNA is double-stranded. Okay, so that's difference number one. So kind of make yourself a side-by-side comparison chart. And start with RNA is single-stranded, DNA is double-stranded. Make your charts correlate with each other. All right, notice they have both of them have bases, but there's one difference. This last one on the RNA side is uracil. So it has CGA, but it doesn't have thymine. So you need to say RNA has uracil instead of thymine. DNA has thymine instead of uracil. And that's important. That way you can, when you're looking at, if somebody gave you a strand to look at, you would be able to say, I know this is RNA because it has uracil. I know this is DNA because one of the bases is thymine. So that's an important structure difference. And again, RNA can leave the nucleus. DNA never leaves the nucleus. So RNA will be found out in the cytoplasm. And so we're going to look at this list over here on the side. There are 3 types. So you need to write these 3 types down. Okay. Type number 1 is messenger RNA, and then they're always abbreviated with the initial in front. So mRNA is messenger RNA. That's the one that can go in the nucleus, that you'll find in the nucleus. And that's the one that is reading the information from the DNA. All right, so again, the boss stays in the office. He shows the messenger RNA all the blueprints. It makes a copy. And then the messenger RNA does what messengers do. It carries it to where it needs to go. So that brings us to number 2. There's a ribosomal RNA, which means these RNA are found in the ribosome. So when we talked about our cell structure, one of the structures that you talked about were ribosomes, and its function was the protein factory. This is where all the proteins are made. So the ribosomal RNA goes to the factory, and it lays out the blueprints for what needs to be made. And then the messenger RNA comes in and reads, actually, I got that kind of backwards. The messenger RNA brings it to the ribosome, and the ribosomal RNA will create a table for the message to be translated. So we're going to be talking about 3 words, translated and transcribed, later on. Okay. All right. And then there's a third one, the transfer RNA. That's the one who's like another little messenger, but he's out in the cell, and he has to go get the parts to assemble together what the instructions say to make. Okay, in this case, we're talking about amino acids, because remember we said amino acids were our units for making proteins. So the transfer RNA goes and finds all of the little amino acids, brings them together so that they can be hooked together in the sequence that the messenger RNA and ribosomal RNA tells it to do. Okay, that may sound a little complicated, but you'll see what it looks like once we start working on it in the lab activity that you're going to do tomorrow. All right, so tomorrow you're just going to build a DNA. So we're just going to be looking at building that. Then we're going to go take the second step to that building DNA after we learn about protein synthesis, and then see how that template is filled in. All right, what I need you to do now is this step right here. So take this information up here and take a few minutes, I'll give you 5, to drag these into the correct boxes. So again, 5 minutes. We'll stop sharing for a minute because I want to check on something. Remember, you're just dragging them into the columns that they need to be in. And technically, what you're dragging should represent what you wrote down earlier. All right. And I'm looking at the assignment for the case of the haunted house, haunted DNA story. And half of you have not done it. Today's actually, okay, it's due tomorrow, but tomorrow it'll be a 0. So you want to make sure that gets in today. All right, that was not extra credit. All right, and I do want to point out some things for those of you who haven't done it already. You are giving a process answer to each of these. You're not giving a general, you're trying to solve a case. We want to know, what do we need to do? How do we solve this case? The first one says, research the process of preserving DNA samples, especially historic ones. So tell me the process. What do we need to do if we are studying an old DNA sample? Second question says, explore the methods of extracting DNA. So one, we have to preserve this specimen when we get it. Secondly, there are things that we have to do to the hair sample to get the DNA out. What do we have to do? A process. Third question, want to know, how do we check the descendants, people who are living today? This person was maybe 100 years old. But today, how will we check people that we suspect might be descendants today? What do we need to do to them to get their DNA sample? Okay. And then lastly, how are we going to use that information to solve the case? Okay. Again, step by step. So I need some steps. All right. So I'm coming back to the module. Okay. So I'm sure hopefully you've checked your answer by now, but what you should have is that DNA is double-stranded, RNA is single-stranded. DNA has deoxyribose sugar, and you need to know that that's a sugar. Ribose is the sugar that you find in RNA. So the D in RNA, the D in DNA stands for deoxy, and the R is for ribose. All right, you will find thymine only in DNA, and you will find uracil only in RNA. And there are 3 types of RNA, and the DNA is only found in the nucleus. Okay, so you should have set yours up like that. All right, and that kind of goes along with the topics down here. Okay, this number 2, you need to be familiar with these terms, purines and pyrimidines. You need to know that adenine and guanine are purines. Now, they are the purines, and then she also mentioned that a purine is always bonded to a pyrimidine. So adenine and guanine are purines, thymine and cytosine are pyrimidines, but adenine pairs with thymine and guanine pairs with cytosine. That is a rule. Okay, and because they pair, yes, there will be an equal amount of each in any DNA sample that you find. In other words, you're not going to have 20 adenines and only 15 thymines because they have to pair up. So if you have 20 adenines, there's going to be 20 thymines as well. Okay, all right. And then we just talked about there are 3 different types of RNA. Okay, those of you who are finished, please take a look at the next one. Those of you who did not write your story, use this last few minutes to make sure that you upload your story. All right, so again, tomorrow we are going to do a gizmo lab, and you'll have to construct, and there's a quiz in it and some other activities. So I'm gonna go ahead and upload, while you're doing this, I'm gonna go ahead and upload that activity so we can, have you all done Gizmo before? No, okay. I'll actually go through the steps with you tomorrow, but what you want to do, there is in your Clever panel, you're going to find something that says Explore Learning. Okay. And when you click on Explore Learning, you may have to log in, but you're already going to be in the system because your names are already in the Gizmo program. All right, so this one's welcoming me, but when you log in, it'll welcome you. And you want to click on the G. And yeah, let me walk you through this today because tomorrow is. All right, so thought I assigned it. Okay. Yep, there it is. All right, so when you log in, what has been assigned to you, there's two things. We didn't do the meiosis one, but we are doing the building DNA one. So you'll click on building DNA. And then we're going to launch it. All right, and then I'm going to send you a handout also for you to answer the questions, but you're going to create, and with this, there's no right answer. You create it in whatever sequence you want to create it in. So in other words, I can drag an A, a T, C. This, you don't, or you could drag them all one by one. So, but doesn't matter. You can repeat them in what other sequence you want to repeat them in, and it doesn't matter. Okay. All right, so that was step one. And then on the handout, it's going to ask you some questions about what you just did. Now, the second thing it's going to tell you to do is to create a complementary strand. Notice it started at the bottom. Why does it start at the bottom? Somebody type in the chat. Why does it start this yellow box at the bottom? What did you learn today that tells you why this one is starting at the bottom? And what do we call this? Somebody type in the chat. Why is this thing, and it's going to be a question they ask you tomorrow. Why is it starting from the bottom? In other words, if I tried to do this up here, that's not going to work. All right. Thank you, Piper. She said that it's going to make an inverted version of the coding strand. So you have a coding strand and an antisense strand. So we're going to start from the bottom and build up. Now, the other thing I want you to notice, base pairing. Notice the shape of these. So they made it easy in case you forget. If you tried, like I just tried to match an A with an A, but notice they don't match. It's like puzzle pieces. They're not going to match. So you forget where it goes, find one, and it's not going to let you do, they want you to do it in order. So you can't just put it there. So let's start with this one. Find something that has a rounded groove on it, and that will match. Okay, good. Thank you, Malik. Good, good, good. All right. I don't know why that just popped up like that. There you go. All right. So tomorrow, that's what you'll do. So if you want to, if you've logged in already and you want to play around with it, go ahead. There is a quiz that you'll have to do. So again, if you're already in and you want to do the quiz, you can do that. The quiz is going to log register in Gizmo for me, but the handout that I'm going to put in for you to work on, you're going to have to send that back to Canvas. So if you have time, you can play with this today, but this is really for tomorrow. Those of you who did not turn in your essay, you need to work on that first. All right. So tomorrow when I give this to you, I'm not going to go through the explaining part because I've just explained to you what to do with it. Okay, so I'm going to come out of this right now and I'm going to upload that worksheet. Was there anybody who had a hard time, was not able to get into Gizmo? Everybody's name should already be in. All righty. Okay, so I really only have 3 people who completed their essay. Again, this is the last week to work on. So several of you were asking what happens if you don't have a printer. The color thing is an extra credit assignment. I will just give half point if you can only upload the answer sheet. So if you can't print it, color it, and photograph it, then because you do have to print it in order to color it, unless you have one of those magic art computers that allow you to color with on the page. If you are not able to color it, at least send back the answer page with the correct answers circled, and I'll give you half credit for that. Again, this is half, these are extra points. Yes, yes. So if you have an art app that you can color virtually, that's wonderful. But everybody, or is that something you can download? Is that something anybody can download? Okay. Oh, there you go. If you want full credit, download the art app and then you can do it like paint by numbers. All right, so that's not due till the end of the week. Progress reports go out next week. So that's just something extra to help out. It's not required. So if you don't do it, I will just blank out that space. So it will not show up as a 0. It just, because the way they are doing it this semester, excuse me, is total points. It just gives you more points or less points if you don't do it. All right. So if you're good, I'm good, and I'll see you tomorrow.